Good morning and welcome to another session of ASM Live at ICAC, coming to you from Chicago. This is the annual infectious diseases meeting of the American Society for Microbiology. I'm Jeffrey Fox, the host of these sessions, and I'm the features editor of Microbe, the monthly magazine of the American Society for Microbiology. A few housekeeping rules for those of you watching us on the internet. If you wish to ask a question, use hashtag ICAC. For those of you in the room, I'll remind you again that raise your hand. Someone will bring you a microphone and identify yourself by name and affiliation. In this session, I have a single guest who is Bruce Weniger, the Associate Editor of Vaccine, and he's going to be talking with us about new vaccine technologies, including needle-free vaccines. I guess let's get started. I'll ask you to give a brief summary of your presentation, and then we'll have a discussion about some of the issues associated with it.
Yes, well, I'll be focusing on alternative ways to deliver vaccines, and one of the major rationales for that is the well-known risks of traditional needle and syringe delivery. In the developing world, there's a lot of unsterile reuse of needles and syringes. In developed countries, there are needle stick injuries and accidents. So we're looking for ways that not only can be safer to immunize, but also perhaps be swifter for use in mass campaigns, and more convenient and simpler to use. And a good example is that many vaccines in the developed world come in pre-filled syringes, which take up a lot of space in the cold chain, and yet in many developing countries, the cold chain is extremely challenged for holding all these wonderful new vaccines. They just don't have enough refrigerators available, and so if we can figure out ways to produce vaccines in much smaller packages that take far fewer cubic centimeters of volume per dose, that would be helpful.
So so tell me what's new and what are some of the things that are stirring excitement? I guess people would be interested in needle-free vaccines as something that would help to popularize vaccines with small children,
right,
and some adults, for that matter.
Well, clearly another rationale for needle-free is the needle phobia that many children and adults as well have, and perhaps that might make them more amenable to coming in to being vaccinated. Plus, in our immunization schedule, we have far more shots than there used to be 10 or 15 years ago because of all the wonderful vaccines that are coming along to protect them against more diseases, and yet children are feeling like pincushions, and parents as well, when they have to get perhaps 4 or 5 shots at the same session. One exciting development is that a microneedle has just appeared on the U.S. market to deliver the influenza vaccine made by the Sanofi Pasteur company. It was just licensed a few months ago in the U.S. It's been on the market for a year or two in Europe already. And this involves just a very tiny microneedle. It's only 1.5 millimeters long, and it barely sticks out, and it's administered directly into the skin, and not entirely pain-free, certainly, because it does penetrate deep enough to hit nerve endings from time to time. But I'm going to be looking for it for myself to see how it feels when I can find it in a pharmacy somewhere during this current flu season. That's one example of a hollow microneedle. There are others that are out there that are in the investigational stage for coating solid antigen or vaccine onto needles that would be placed in a Band-Aid or plaster, as the Europeans or the British like to call it, onto the skin, kept there for a few minutes or longer, peeled off, and the child or the adult is vaccinated because the drug has dissolved from its dried state on those microneedles. And some of these microneedles are so short, say 250 or 300 microns in length, which is like 1/3 roughly of a millimeter, that they won't cause any pain because the nerve endings don't end until much deeper in the skin.
Can you just let me interrupt you for a second, because some people aren't familiar with millimeters and micrometers.
Okay.
Are we talking hair widths, the size of a period on a printed page, or what sort of size are we talking about?
Right. Well, the microneedle size, or the mini needle size of the device I just described that Sanofi is using for its influenza vaccine has an outer diameter of 0.3 of a millimeter. Now, I don't usually work in the English system of measurements, but that's going to be probably about a quarter of a pencil, of the lead in a pencil's thickness. You can just barely see it. And the other ones I've mentioned are really microscopic. You cannot really see them with the naked eye, and they could be painless. Now, there are some other technologies out there, such as jet injection, which are like powerful squirt guns that have been used for over 50 years, invented actually in France in the 1800s, and then used by the military for many years for mass vaccinations, and those who were in the military up until about the 1990s probably received vaccines from these. These were taken off the market because they were using the same front end or nozzle to go from one patient to the next standing in line. As a result of that withdrawal for safety reasons, a new generation of disposable syringe jet injectors came on board, which use a separate single-use cartridge or syringe at the front to vaccinate each patient, and then it's thrown out and a fresh one is replaced. And this, as I mentioned, is like a powerful squirt gun. There's no needle at all, and it just pressurizes the liquid vaccine to put it through a tiny hole like a child's water pistol with enough power to penetrate as deep or as shallow as you wish. And some of these go into the muscle or the fat if that's the desired target. And the new ones that I'll be talking about today are going into the skin because the skin is very richly endowed with immune system cells, and it can often take advantage of using a much smaller dose. So in times of shortage, for example, when we had the shortage of influenza vaccine for either H1N1 in the beginning, or in many developing countries that couldn't afford to use full doses, the concept would be maybe you can get by with 1/5 of the regular dose and protect 5 times as many people as otherwise by putting the drug into the skin.
Now, are there some, I guess there's a lot of reformulation of vaccines that would accompany the use of a new delivery technology. Are there any instances where there's a problem of not being able to deliver enough material, or even knowing how much you're delivering if you're doing a pinprick that nobody feels?
Right. Some of the technologies I'll be talking about, such as jet injectors, use off-the-shelf conventional liquid vaccines, so that the requirements to reformulate them and go through all the regulatory processes and toxicity testing and so forth that would be a barrier for new formulations do not exist for them. So the idea for them is to take the very same vaccine that would otherwise be delivered with a needle, put it into a jet injector, either in full dose into the muscle or fat, or a reduced dose into the skin, and get the same effect if clinical trials approve it. Many of the others I'll be talking about, the Band-Aid vaccines, various patches, microneedles, and so forth, do require reformulation, which is a more expensive process, and they may not be as inexpensive as existing vaccines, at least for some time.
At the beginning. Now, when you say reformulation, are there adjuvants or other additions that become part of the vehicle of the vaccine, so that you have better transfer from the bandage or the from that material into the recipient?
Right. Well, the adjuvants that you mentioned, which in case the audience is not familiar, it simply means a chemical, not the actual vaccine itself, that enhances the immune response to make the vaccine work better than it might otherwise. For the skin, which will be the focus of this afternoon's discussion, one of the key issues is whether adjuvants would be too irritating into the skin. In fact, one reason we prefer giving most vaccines into the muscle is to get it as far away from the skin as possible, because many of them are adjuvanted with these chemicals that cause irritation. In fact, the irritation may be signs that the immune system is responding nicely. So that's one question, whether if you use the intradermal approach to deliver vaccines to the skin, can you also use an adjuvant? There may be some inability to do both at the same time.
All right, so some incompatibility potential.
Not incompatibility in terms of being effective, but incompatibility in terms of being too irritating that patients will reject it if it's leaving a large reaction on the skin.
Well, I was kind of groping for trying to formulate my own question about, are there any other chemicals being introduced here that would help move the vaccine antigen across into the skin, into the right part of into the right depth, or
well, I didn't
because
mention
there is a kind of a tough area of the skin on the top that isn't penetrable, right?
Right. Well, I didn't mention one of the most exciting areas, which may still be at least 5 to 10 years away, are dissolving microneedles, in which instead of being solid and having metal or ceramic, once they're put into the skin, the vaccine has been embedded or encapsulated in the matrix, a biocompatible, non-toxic substance that's, when it's dry, it's sharp enough to penetrate, but as it acquires the moisture from the skin, it dissolves and the vaccine or drug can start to do its job. And then once you've pulled it out, there's nothing sharp left, even a microscopic projection of any kind. And this is very exciting. Work is being done now with animals. I'm not aware of any human trials yet of a dissolving microneedle. But for the most part, all of these devices I have mentioned and will be talking about are required to be sharp enough to penetrate through that outer barrier of the skin. That's the key obstacle to achieve vaccination into the skin.
And is there any role for nanotechnology in these developments?
Well, I think I consider nanotechnology really a separate issue, which is the size of the particles that you're administering, and whether you're delivering a conventional sized particle or a nanoparticle. I think it's a separate question from how you get it into the body, whether it's with a conventional needle and syringe or a needle-free jet injector or a microneedle or a hollow or dissolving version of them. So I think the nano is an exciting area, but I think it's not, and it really has to do more with a new wave of new vaccine constructs, liposomes and other ways of packaging the vaccine, which I, in my mind, keep separate from the actual mechanical process of how do you get it into the body.
All right. No, that's a fair set of categories. I'm curious, though, whether having new ways of delivering the vaccine opens the door to new kinds of vaccines. Does it broaden the scope of diseases and pathogens that you can help kind of count on protecting people against?
Perhaps. I think every time you add a new variable to the equation, it complicates the scientific studies you have to do to demonstrate that it works and it's safe. If you are delivering a conventional vaccine by a new method, that's probably a little bit easier to get through the regulatory authorities than if you are delivering an entirely new construct by an alternative method. Although I think if you're going to start and develop a new type of vaccine that's going to go from day one to prove it's safe and effective in animals and then humans, you probably ought to start with a delivery system that's of the future rather than of the past. So if you have a choice and you're a vaccine company designing a new antigen, it would seem to me to make sense to start from the very beginning with needle-free methods of delivering it, so that by the time you reach the market, you're competing with other needle-free routes that might be out there as well. And I think, needles will be with us for quite some time, but they've caused a lot of problems over the last century and a half of their use, and the more we can get away from needles and delivering old drugs, the better we're going to be.
Now, maybe this is your specialty in terms of trying to replace needles, but there are other routes of administration. There are some inhaled vaccines, there are some orally delivered vaccines. how is that going to shake out in terms of having these new technologies available, but are they going to compete well with other delivery, altogether different delivery systems?
Well, I think these other methods, squirting into the nose, inhaling into the lungs, taking liquids or solids by mouth, are all needle-free, and in that sense they have advantages over needles and syringes. I happen to believe, though, that the cutaneous route, or putting it into the skin, has advantages over all of those other ones, and I can give you some examples. When you put things into the body, there's always the risk of adverse events, sometimes unanticipated and sometimes quite severe. We learned from putting the rotavirus vaccine in the late 1990s into the gut that it caused intussusception, a serious problem, although it occurs quite rarely. If you put things into the lung, what happens if you get a severe reaction at the site of deposition? The lung is an essential organ for life, and it's a lot harder to deal with a reaction or inflammation in the lung than it is on your arm. So I think as the least invasive route, the skin offers the advantages. If you are going to have a local reaction where the drug is delivered, it's easier to treat, it's easier to see, it's easier to put steroids on it to cut down any inflammation. So it has that advantage of being the least invasive route. Another advantage, or let me say, give some disadvantages of other routes. The respiratory route often requires patient cooperation to suck in on an inhaler, or it might require a mask that takes up to 30 seconds to deliver the dose, which can, in a mass campaign in the developing world, can take a lot of time. The squirts in the nose, the trials, I do not believe included children who had what's often been called the 11th sign, a chronic purulent rhinitis or serous rhinitis with the dripping liquid from the nose coming down and being recycled in the mouth. And in the developing world, that's quite common in harsh climates to see children with a chronic rhinorrhea. We don't know whether trying to vaccinate into the nose with the existing influenza vaccine would vaccinate those children successfully. Whereas if you're avoiding all those potential barriers to more invasive routes in the skin, you're pretty sure when you've delivered it that it's gotten in, because you can see it.
All right, well, so correct me if I'm wrong, but so far as I know, the skin is not, wouldn't work as a route for delivering a live vaccine, or is that not the case? I mean, are there some problems, almost categorical problems?
Well, I think there are issues with live vaccines, but you have to remember our very first vaccine that Edward Jenner first published in 1798 was smallpox vaccine, which went into the skin, and it worked by being a live vaccine of vaccinia or cowpox virus that would grow in the skin. There are very few studies that I'm aware, and BCG also is traditionally given into the skin and multiplies in the skin, so you see the reaction from the BCG vaccine of its growth. I'm not aware of any more recent studies on other live vaccines given into the skin. Most of the time they are inactivated vaccines. But I think each attempt to put a new vaccine into the skin that's never been studied before by that route would certainly require clinical trials to prove that it was safe and effective. But two of the most common, and even yellow fever was for many years delivered, a live vaccine was delivered into the skin as well. So there were three examples of skin vaccines, but it's going to be really dependent on the behavior of that virus or a live bacteria on how it's going to work when put into the skin.
All right. We do have a question in the room, Jim, if you could bring the microphone up. A couple of questions.
Hi, Laura Pullen, Clinical Podcast News. I'm interested from an immunological perspective. You touched on this. It seems like going into the skin is fabulous because you're taking advantage of dendritic cells. You actually should be able to mount a response that's even better than what you're getting by doing it IM or other ways. And you touched on that by talking about giving less of a dose, but are you also seeing a better immunological response, better coverage? So that would be number one. And then my other question, Do you think that's going to facilitate creating vaccines against organisms that so far we haven't been able to create vaccines for, because now you're going through this different, perhaps more immunogenic, well, absolutely more immunogenic route?
Yes, well, I think one of the potential, and I use the word potential advisedly, advantages of the skin is that often, but not always, you can get away with using less of a dose than you would have to give into the muscle or the fat to achieve the same effect. And the advantage of this is if you have a shortage of vaccine, if there's a new epidemic threat and there's not enough vaccine to go around, that you might use dose sparing. It's often called a smaller dose to get the same effect by putting it into the skin, but not always. The literature is pretty good for influenza vaccine, although not in all cases, going back as early as 1937 to use reduced doses into the skin. The current Sanofi product in the United States delivers 9 micrograms for each virus strain in its influenza vaccine compared to the usual 15. So it's dose sparing somewhat. I guess that's around a 35. I didn't do the calculation how much it's saving from 15 down to 9. They also have a product not yet on the US market in Europe and other countries that for the elderly that have not so good immune systems, they begin to wear out. And they are using the same 15 micrograms in their intradermal injection that they would otherwise give into the muscle as a means of trying to boost the effect on the elderly who are not responding very well to influenza vaccines at all. And so they're not dose sparing at all, but they're giving the same dose in a smaller volume. They're concentrating it down to 0.1 ml, which is the traditional intradermal volume. Because you couldn't, easily put 0.5 of an ml into the skin. But that's not yet on the US market. And if you look at the numbers, which are in my slide for this afternoon, they are getting better responses to 15 in the skin in the elderly than 15 micrograms per virus strain per dose when delivered into the muscle.
We have another question from the room.
Ed Sussman, Medical Front Page. A lot of what you're talking about would relieve so-called needle phobia from vaccines, but how would this fit into relieving vaccine phobia, which appears to be a new, or relatively new problem that we're facing? Is needle phobia somehow connected to vaccine phobia, and in some way, or is vaccine phobia something totally different?
Well, I think you raise an entirely different issue. We've seen it in some of the presidential debates and recently. I don't consider myself an expert or an authority on the anti-vaccine movement and the parental fears of vaccine, but I think my colleague, formerly at CDC when I worked there, has illustrated this well with a graph in which he says, as our vaccines become so successful and eliminate the diseases from the population, the young parents who are the ones who have children no longer recall a child across the street getting measles or a child down the block dying of whooping cough or another serious disease. And they think, well, these diseases, they're not a problem because they haven't seen them. And my suggestion to parents who are wondering about whether these vaccines are safe is to think about whether the diseases are safe and to go to YouTube and type whooping cough or type pertussis. You have to figure out how to spell it correctly and see these video clips from not only from the 1950s, but even from the 1990s or even the 2000s of kids in modern intensive care units whose parents neglected to get them vaccinated against whooping cough and other diseases, and see how they suffer. And for, in the case of pertussis or whooping cough, they go for weeks, if not months, struggling to get a breath in ICUs. And so I think it's ironic that as we're more successful with vaccines, we eliminate the diseases, the parents don't realize what they were preventing by these shots, and then they let their kids go at risk for getting the diseases.
Is there a lot of times parents who have children who are vaccinated with current vaccine needles, the children get very upset, cry, scream, yell, and is that part of what later becomes vaccine phobia because the parents don't like to see their children crying?
Well, 10 or 15 years ago, and I've spent the last half of my CDC career involved with this, I sort of felt that the increasing number of new vaccines would cause a problem. And one of the ways they've solved that was by combining the vaccines. Instead of having to get 2 or 3 separate shots, they'll put 2 or 3 separate vaccines into one shot. And that has helped, but it's a very expensive process. It takes years of development and costs hundreds of millions of dollars, and it often doesn't work. That one vaccine in the same syringe will interfere with the take or the success of the other and reduce the response. But that is one way that it's been solved. But we still have a lot of shots, and I think I'm not privy to the marketing strategy of Sanofi Pasteur, but I suspect one of the reasons they bought this technology called the Soluvia, the micro needle that delivers the vaccine into the skin, was to distinguish it from their competitors, and maybe it was certainly quick and easy to deliver, and maybe they would see some benefit from in terms of market preference for it. Now, over the years, I've been responsible for oversight of about $15 million in CDC contracts for small business innovation research that we felt was relevant to public health needs. And we've provided funds to a number of these small companies to develop new generation needle-free jet injectors. And one of the companies that we worked with was the PharmaJet company in Golden, Colorado, that got on the market, licensed their jet injector, not the intradermal version that's still investigational, but actually I think the intradermal one was licensed this year by the FDA, but the one that goes into the muscle. And they were using it during the 2009 season in county health clinics around the country. A lot of counties around the country were using this technology to deliver the routine seasonal influenza vaccine. In fact, when I was in a supermarket in that year, they were using that technology, and I tried it out myself, and it felt fine. Not a useful, just an anecdote certainly, but I think when the public was asked, we can give you the shot with a needle and syringe or with this needle-free device, a substantial number were choosing the needle-free device, and there were no complaints to my knowledge about that choice. So I think there may be some competitive advantages to needle-free that might help vaccine companies market their products compared to competitors still delivering it with conventional needle and syringe.
Hi, Charlene Lena with WebMD. With the Sanofi Pasteur flu vaccine that you talked about that's just on the market, did that have to be reformulated, and what was required for it to become on the market? I mean, did they prove that they got an equal dose, and can you describe whatever studies were done of it?
Yes, and I'm not aware of their actual detailed manufacturing processes, but they clearly had to concentrate the vaccine from the usual volume of 15 micrograms of virus per strain per half of a cc or half of an ml down to 0.1. And they've also reduced the amount of the antigen. So that was technically a reformulation, and I'm sure it's gone through all the studies and regulatory approvals that it needs. Whether they're using the same original batches of their flu zone vaccine and simply changing the amount of dilution, that's proprietary information. I'm not familiar with it. But my guess is it's probably the very same vaccine that you can also buy with a needle and syringe, but they've simply used less water to dilute it to achieve the desired concentration.
But, like, what was required, if anything, in terms of studies? I mean,
they've done probably at least a dozen or so studies in Europe and America for this technology, and some of them may not be published, and some of them may only be shared with the FDA or the European authorities. But there have been a number of published studies on the delivery of this device. In fact, you can look at their product insert, which I'll be showing some data from this afternoon of what they found when they compared it to the regular dose with needle and syringe.
Okay, let me ask you about mucosal immunity. Experts who are developing vaccines and are concerned about the immune system often speak about the challenges of inducing mucosal immunity. How this seems to not to address that, and I just want to make sure that, or is there a way where this might improve matters?
Well, I would consider mucosal immunity to be delivery into the mouth for either ingestion or swallowing the dose, or into the nose for beginning of the immune response. There are other orifices of the body that have been used, but I think they're not practical and are not going anywhere beyond the mice or the primates that they've been studied in. And I think it's fine. We've had the oral polio vaccine since the 1960s, and it's been very successful in almost eliminating polio from the world. And one of the things I'll be mentioning this afternoon is that that vaccine being live has problems when, quite rarely, it can revert to virulence, and the very vaccine virus itself can cause polio in very rare cases. So eventually, and relatively soon, the polio eradication program wants to take oral polio vaccine off the market and replace it with the injectable killed polio vaccine. The problem is that vaccine costs 10 times as much per dose. So one strategy that WHO is considering is the intradermal route, and I'll be mentioning that this afternoon. If they give 1/5 the dose, the normal dose of the injectable polio vaccine, will they get as good a response as with the full dose? And two studies have been published to date, and if the vaccine is given at 2, 4, and 6 months of age, the answer is yes. If it's given earlier than that, one study didn't show as good a response, but more studies are underway, and they'll be using needle-free jet injectors delivering vaccine into the skin. That's one of their major delivery methods for these ongoing studies. Another one is an adapter for a BCG or tuberculin type syringe that makes the regular intradermal injection. That's called the Mantoux method, after one of the French doctor who invented it 100 years ago. That's very difficult to do, but this new adapter that actually CDC helped fund its research and development makes it quite simple. You just push it in and squeeze the plunger down, and you get a wheel every time, which if you ask nurses and doctors, that's not very common with the conventional way of delivering intradermal injections.
Okay, and there again, though, with the polio virus or polio vaccine, another part of the change is moving from a live oral to a killed viral vaccine, but trying to get a higher immune response from that killed vaccine.
Right. There's a lot of work going on with new vaccines for oral delivery. Intranasal delivery, respiratory delivery. That's not the focus of my talk today. I've focused it down on cutaneous.
And I wanted to follow up also just briefly about, you mentioned the rotavirus vaccine and pointing out that the early version of it was delivered orally and there were problems associated with it. So the vaccine that's now in use is an injectable, is that right? Is that,
No, the two rotavirus vaccines.
I know it was reformulated as well.
Yeah, they are still both delivered orally. And a session I attended just an hour ago downstairs discussed the fact that the occurrence of intussusceptions is still occurring, but at an extremely rare rate. From Mexico, I think they found 1 in 60,000 cases of intussusception. However, they also found that the deaths prevented from the diarrhea caused by the rotavirus greatly outweighed the occurrence of the intussusception. So from a benefit analysis, that you're saving thousands of lives and causing intussusception in a few cases was worth that trade-off.
All right, but there's no plans afoot then to reformulate or reconstruct that vaccine so that it could be delivered in the way you're discussing?
No, I don't think so. I don't think they've earned yet back their investment costs for putting it into the oral delivery route. But one of the challenges for rotavirus vaccine is it comes in pre-filled syringes, not for needle injection, but for oral instillation. And these are taking up a huge volume in the cold chain of developing countries that could definitely need this vaccine. So there's a lot of pressure to try to reduce the volume per dose required for cold chain preservation until the vaccine can be delivered.
All right. And the other possibility, sort of a future scenario, is that a whole slew of injectable childhood vaccines could be delivered in a less painful, less frightening or anxiety-provoking way, and they might all be sort of condensed and put together, or?
Well, I don't think I could categorically agree that all existing vaccines could be delivered with these newer methods. I think each and every one has to be demonstrated with reformulations and safety studies in animals and then clinical trials in humans, but they are promising. But some of these methods that I've described, such as the jet injectors, the adapters to make intradermal delivery with a syringe, can use existing and are using existing off-the-shelf vaccines. So the same vaccine that you would otherwise give with a needle and syringe, you can give with a needle-free jet injector.
All right, so, and flu vaccine seems to be the most advanced in terms of this approach to delivery, or
well,
are there other leading candidates?
There are quite a few other vaccines that are being studied. I mentioned flu because it's now on the market in multiple countries for delivery into the skin. And of course, BCG vaccine, not used in this country, has always been delivered into the skin, and the U.S. military and other militaries around the world that continue to vaccinate against smallpox for concern of bioterrorism are still delivering into the skin. So there are still other licensed vaccines going into the skin, but most of them are not. But examples of others, one of the new dengue vaccines is being researched for delivery into the skin with one of the jet injectors that I mentioned. And there are others as well, hepatitis B. Influenza is quite popular because it's a vaccine that's recommended for annual use all the time, and so there's a huge influenza market. So I think that's what's probably driving vaccine developers to put that antigen into a needle-free system.
Okay. We have a question from Jim.
Yeah, actually we have a question from the internet. One of our viewers on Ustream wants to know what are the main challenges to produce a vaccine for a virus with antigenic variation?
Well, that's a broad question. I'm not sure it's really one that I feel comfortable answering. It's not really related to how you get the vaccine into the body, and I'm not sure this is the appropriate forum for me to try to attempt to answer that, but variation has been raised with the effectiveness of some vaccines because it's been hypothesized that the wild bacteria, wild viruses out there that are circulating are changing and no longer match the bacteria or virus that was used to make the vaccine. And if there is that mismatch, there's the theoretical possibility that you won't get as good a protection as was found during the clinical trials when the wild virus or the wild bacteria strain that was out there was studied, was the threat against which the vaccine was originally demonstrated to be effective.
Okay, any final comments, final questions? Do you have any summary statement you'd like to share, or something perhaps forgotten?
Well,
until this moment?
We've been working on these needle-free systems for 15 years, and for a number of years I've been quite pessimistic that the field was not going anywhere. And now I'm really quite optimistic that there's a lot more interest in these alternative routes for a variety of reasons. One of them, I think, was the dose-sparing concern, and we have had shortages of both the seasonal influenza vaccine and delays in getting the H1N1 out when we needed it in 2009. And with the new movie Contagion, there's always going to be some new vaccine that will be coming, that'll be threatening the human population, and if...
No, wait a minute, not the vaccine threatening the human population.
Oh, no, the disease threatening the human population. But certainly there will be shortages, as we saw in the movie, of when sufficient supplies can reach the entire population. And dose sparing, being able to use smaller quantities of vaccine if it's delivered into the skin, might be one strategy in those early months. In fact, I can't remember whether I think the vaccine in the movie was they were using the Becton Dickinson AccuSpray, the same method of squirting into the nose that is used for the licensed MedImmune influenza vaccine flu mist. But whether or not kids with the 11th sign would be successfully vaccinated with this hypothetical new vaccine is an open question. So the skin might be another route for the next movie, the follow-up Contagion 2, maybe.
Contagion 2, it's not coming to your theaters anytime soon, but look forward to it. But we have been talking about new vaccine technologies, and for the second day in a row, renewed optimism among microbiologists about combating infectious diseases. This concludes our session, 11 o'clock session, New Vaccine Technologies. I've been talking with Bruce Weniger, an associate editor of Vaccine, the journal Vaccine. Thanks very much for joining us. We have another session in about 15 minutes.
Thank you.
Thanks.
Thank you.